All right, how you doing today, Fred?
I'm okay.
So we're going to talk today about a fluorescence experiment. Now, let me see what you know about fluorescence.
Well, it's kind of like absorption.
That's right. It's a spectroscopic technique where you shine light on the sample, but how is it different from absorption?
Well, the light, you just measure the light that's emitted by the sample after it absorbs the energy that you shine on it.
Okay, very good. So we have our sample, right? And our sample, which we'll draw in a little cuvette here. We have a light source, right? And the sample absorbs some electromagnetic energy from our source, right? And if we were to measure absorbance, we would look at how much light was leaving the source, how much electromagnetic energy was leaving the source, and compare it to how much was getting to the detector. If that would be absorbance, right?
Yeah, but fluorescence goes, you look at a different angle.
That's right, because as the chemical in here, the analyte in here, absorbs that light, oftentimes it will re-emit photons of energy of a different wavelength. And what did we say the relationship between the wavelength of light that is absorbed compared to the wavelength of light that is emitted?
Um, it emits lower energy, so that means it's a lower frequency, and it has a longer wavelength.
Very good. So then we have a detector at a different angle, like at 90 degrees or something, and we're not looking at the light that's what's being absorbed. We're looking at the photons that are being emitted or fluoresced after the electromagnetic energy is absorbed. Okay? And in this technique, or this experiment, this type of experiment, we're going to be looking at proteins because proteins frequently have fluoresce. They frequently fluoresce. I mean, all proteins probably fluoresce to some degree. Well, unless they're really small and they don't have the amino acids that are tend to fluoresce or have fluorescent properties. Okay? Now, let's make sure we all understand or on the same page or figure out where we're at. You said, what did you say about biochemistry?
Yeah, I had some of that.
Okay. So do you remember how proteins are made or what proteins are made of?
You mean like the ribosome and stuff?
Well, not how they're made, but what are they chemically composed of?
Carbon, nitrogen, hydrogen, oxygen.
That's right. But what do we call the units that make up proteins?
Oh, amino acids.
Right. Very good. Amino acids. Now, are you familiar with any of the amino acids?
I remember histidine. We talked about that earlier. Proline, alanine, phenylalanine.
Okay, good. So you have some. Let's bring up a table of some of our amino acids. Do you remember how many there are?
Oh, there's 20.
Okay, so here's our amino acids that make up our proteins, right? And do you remember how the amino acids link together?
It's called a peptide bond. I remember that.
All right. So you see how each amino acid has a double bond here and a hydroxyl group here, which that combination, carbon with a double bond and a hydroxyl group, is called a carboxylic acid. And then this next carbon down here has an amino group here. Now, proline's a little bit different in that the amino group is actually part of the cycle here, but all the rest don't do that. And then we have the carboxyl group and what we call the amino group, and that's why this is called a carboxylic acid again, and so that's why they're called amino acids. Okay. And then you have the rest, or what we call the side chain, okay? And the 20 different amino acids differ in terms of the different kinds of side chains that they have. When two amino acids come together, the way that they do it is the electron density on the nitrogen reaches out and grabs onto this carbon. That carbon is poor in electron density. Any reasons why you could imagine that that carbon would be poor? I mean, would be slightly positively charged?
Well, it has two oxygens on it, and those are electronegative, so maybe the oxygens are pulling the electron density away.
That's right. This oxygen and this oxygen are pulling electron density away. Now, let me remind you also of something that we talked about not too long ago. Let me get a scratch paper because this is just going to be a small little image. But remember that when we see something like this, a carboxylic acid group, right? Or meaning just the rest of whatever's down below. This group can also be kind of drawn like this, where you have this idea of a resonance structure and negative charges on both oxygens. Does that make sense?
Yeah, okay, I remember that.
So we don't want to think of it really as an asymmetric system. It's symmetric. I mean, it kind of looks like it's asymmetric because you have a double bond here and a single bond oxygen with a hydrogen. But that hydrogen can jump back and forth to this oxygen very easy, and it doesn't have to be the same hydrogen. This hydrogen can be pulled off by water, and a different water molecule can donate a hydrogen here. So that double bond is going back and forth. So it's very symmetric in terms of their properties of those oxygens. So you have then a peptide bond forming when this nitrogen from one amino acid attacks the backside here, comes around the back, and attacks the carbon on the carboxylic acid. And that one of these hydrogens leaves when that occurs, because it doesn't need to have, right here it has 3 bonds, it doesn't need to have the fourth. It can have the fourth, and it depends on the pH exactly. But usually at a higher pH, where there's more basic conditions, that hydrogen will be pulled off, extra electron density then if that proton is removed. That positively charged electron density, negatively charged electron density attacks this slightly positively charged carbon, and at the same time this oxygen or hydrogen will dissociate from this oxygen, this whole group here, this OH group will dissociate from this, not dissociate, but actually be removed from this carboxylic acid group. So let me draw that chemistry a little bit better so that we can make sure we all understand what's going on. So you have a carboxylic acid group. This carboxylic acid group has, this is an amino acid again, so we'll draw R for the rest, the side chain, right? Does that make sense?
Yeah, I get that. And the side chain, there's lots of different things that the side chain can be, right? Like 20 different things.
Yeah, see all these different kinds of side chains?
I noticed it says 21 amino acids. I thought you said there was 20.
Well, it's interesting. They're including selenocysteine here, which, I mean, I'm not sure about its natural abundance, natural occurrence, but yeah. So they have a list of 21 here, but yeah, I don't know. It'd be interesting to find out if selenocysteine is truly contributed significantly in nature somewhere. But you can have an infinite number of amino acids, right, by just changing the R group. These are just the 20 amino acids that are that nature uses to make proteins, right? So you can do all sorts of chemistry and have this functional amino acid component, which would make it an amino acid. Okay, so amino acid 1 here, and the second amino acid is going to create a peptide bond. So we're going to get the nitrogen of a second amino acid and the carboxylic acid, whoops, that's not very good, carboxylic acid of the second amino acid, the R group here, NH2. So electron density from this nitrogen comes and attacks right here, and electrons go on to the oxygen there. This is generally going to be received by a water molecule or a proton that's in solution. So if you have slightly acidic conditions here, then the electron density will pull over to here. right? And so what results is a peptide bond, which is going to now have this double bond bound to this nitrogen here. So I'm going to call this nitrogen 2. This is nitrogen 1. Here's the carboxylic acid carbon 1. Here's the carboxylic acid carbon 2. Here's the nitrogen carbon 2. Here's the amino acid carbon 2. Here's the amino carbon 2. Sorry, this is amino carbon 1. All right, R group 1 will label R group 2. Okay, so all of this here is our first amino acid, and all of this here is our second amino acid. Okay, so in the end, we're going to have this carbon now attached to a nitrogen, right? And this is going to be our other carbon here with the nitrogen. I'll draw these hydrogens out. This is nitrogen 1 now. This is carbon 1. This is the carboxylic acid carbon 1. This is the oxygen from the carboxylic acid carbon 1. This is the nitrogen from the 2. It's connected to a carbon. All right. And then that carbon has the R group 2 coming off it. This carbon here has R group 1 coming off of it. All right. This carbon 2 is connected to this carboxylic acid carbon 2. Okay, 2, 2, and the 2 there. All right, so that's our peptide bond. The peptide bond is an amide bond. And do you remember there's a special property about the amide bond that we always want to remember. Do you remember that from biochemistry?
Does it have, not really, no.
Well, it has to do with protein folding, and it limits the way that a protein can fold, because this functional group, the amide functional group, is, and I should have drawn one more hydrogen here, is similar in resonance structure to a carboxylic acid, right? In that the electron density from this oxygen often is brought down here. So if I were to redraw a oxygen nitrogen amide group, it would be reasonable to think of it like that. So electron density is being shared across this bond, not just in a sigma bond, but also in a partial pi bond, which prevents this bond from rotating. All right, so you don't get rotation here. You can have free rotation here or here, but not here. And then the next bond here would be a nitrogen bond if you're going to have a third peptide on here. So you have two bonds where there is free rotation, and then a third where there's not. Two bonds would have free rotation, and the third would not. And so the amino acid chain follows this pattern as it goes along, right? So you have a nitrogen to a carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, all right? One amino acid, two amino acids, 3 amino acids, 4 amino acids. So let's say this is our peptide bond here, right? So nitrogen, carbon, carbon, nitrogen, carbon. Our second peptide bond would be there. I'm going to put a 1 here to say that's the first peptide bond, second peptide bond there. The third one would be here, okay? And the fourth one would be there, okay? So you have, again, free rotation here, but not free rotation there or there. Free rotation here, but not free rotation there. And so this limitation limits the number of configurations that a protein can fold into. Now, do you remember kind of the groups that proteins fold into?
What's it called? Alpha helix?
That's right, an alpha helix group. That's right. So, we just kind of draw that like this sort of thing, where this chain is really a chain of amino acids, right?
Yeah, and then there's the beta sheets as well.
Very good, beta sheets, where the strand of amino acids will kind of bend along themselves like this. Now, when this occurs, you get interaction above and below the helix and to the right and to the left in a beta pleat or beta sheet, right? And what those are usually hydrogen bondings or other kinds of interactions, because you have lots of oxygens here and you have lots of nitrogens. And so when oxygens on one side can interact with nitrogens on the other, then you can get a strong interaction, intermolecular interaction. It's really an intramolecular because it's a really big molecule, but it's, you know, it's an interaction that's bringing two portions of the molecule together. Okay, so if we have a protein, then here's my protein that I'm going to draw. All right. It's a strand that folds, right? So if we wanted to kind of suggest that, all right, this comes out of the ribozyme, or the ribosome, right? Comes out of the ribosome like a long strand and then folds gradually as it's created. It does that. But if we could think of it as a folding, maybe an unfolding equilibrium, right? This folded version is the active version. It performs its whether it's a catalytic property or if it's, you know, whatever function it has, the form here determines its functionality. And the primary amino acid sequence governs the secondary secondary structure, which would be alpha helix or beta pleats. And then how those structures would come together would be your tertiary structure, and then your final quaternary structure would be how smaller proteins come together to form larger complexes. Okay, so we got proteins that are made of what?
Amino acids.
That's right. So here are our amino acids. Now, chemicals that we talked about this a little bit ago, but what kinds of chemicals can absorb electromagnetic energy? Or what kind of chemicals can absorb an increased amount of electromagnetic energy?
You mean like aromatic and stuff?
That's right. That's right. Aromatic chemicals like these regions down here, phenylalanine, tyrosine, and tryptophan, right? Anytime you see that sort of a ring with multiple double bonds, right? This is called, you have a conjugated system here where you have a double bond every other bond, and so you have a free pi orbital there, and the result is the electrons begin to be shared, and it behaves more like a molecular orbital. And these molecular orbitals then have the ability to kind of absorb energy in more regions than a simple chemical like alanine, right? So phenylalanine, tryptophan, and tyrosine are our amino acids in proteins that cause them to fluoresce. And again, what that means is these molecules absorb energy and then re-emit that energy in the form of a photon, and it's a very particular wavelength. Now, let's look at a protein here. So here we have a protein. It's a long chain protein. This is bovine serum albumin. It's just a very common protein that is used in biochemistry, usually just for a kind of a control. Here's where it starts, the blue region over here, and here's the other side where it ends. And I'm not sure, but it looks like this is not a dimer. Looks like this is just a single strand that runs through. Oh no, there. Let's see. It is a dimer. It is a dimer. Okay, so we have two identical units, and that's typical, right? Typically, proteins either come as dimers or tetramers, sometimes trimers, and complexes, which are not necessarily always symmetrical, but they'll have multiple proteins. But so here we have a dimer, And if I want to find out where all the fluorescent amino acids are, right? And I'm going to select certain residues. Residue is another name for amino acids in the protein, right? Let's go with the tryptophan. That's generally the one that is kind of thought of as our fluorescent ones. And then I'll say show here, and I can see these regions that have shown up. See, here's the side chain of a tryptophan. So what I did was I selected all the tryptophans and said, show their side chains for me, right? So I could hide them and then I could show them again. So we can see where the tryptophans are, right? So it looks like we have, is it just 2 tryptophans? All right, so one here and one here. Okay, this one here is pretty close to the surface. It seems to be a real good access to the surface there. Where's the other one at? There's the other one. I would say this one looks like it's, whoops, what's going on here? Why is it doing that all of a sudden? Oh, brother. There we go. So this one here, I would say it's a little more buried, right? Can you see that?
What do you mean?
Well, let me take this whole protein here and project it as first, let's take those amino acids there that we selected, and we project them as a kind of spheres, as where their electron density would be. And then if we select the whole thing and do that same thing, make it all spheres. Show, there we go. Okay, so now we got the molecule. Let's get rid of its glow. Is that going to get rid of its glow? Select, oh, all clear, there we go. There we go. So, here's what our protein would kind of look like if we could see it, right? And this is the electron density that your X-rays kind of diffract off of, and that's how we know this structure, from X-ray diffraction. Okay, so we have this crystal here, or sorry, we have this protein here, and now let's see if we can kind of select the residues again. Let's select our tryptophan. Let's see if we can change their color. So now, well, you can see the kind of glow right here, right? So this is the one that we said looks like it was kind of accessible. All right, let's see if where's that green one at? Yeah, see that green one's really buried in there. Not a lot of accessibility. Okay. So that accessibility will change the ability for it, or its capability to fluoresce, because it's altering its capability to absorb light, right? So if the light, it gets kind of diffracted or absorbed by other proteins here that don't fluoresce necessarily, then that tryptophan in there is not going to fluoresce as well. So in other words, there's a difference in the amount of fluorescence between tryptophans that are kind of near the surface, where they're going to be hit by electromagnetic energy more directly, than those that are buried. All right. Now, let's go back to, let's see, let's select all again and go back to the stick mode here. And, oh, not to the stick mode. We want to hide that. All right. So we have the protein again here. This time, I'm going to just select the residues. We'll go back to the tryptophans again. And we'll also, so I'm going to show those. So they're back here now. Now I'm going to select also the other residues. Let's look at the tyrosine and show those. Okay. So a lot more tyrosine. Do you see all that tyrosine?
Yeah.
Now tyrosine does fluoresce, but not nearly as much as tryptophan. In fact, you know, if you want to have a protein really fluoresce well, people will genetically alter it and put a couple of tryptophans on there so that you can learn about the protein's kind of structure. Because, like we said, if the tryptophan is buried, it'll fluoresce differently. So if the protein moves, the fluorescence will change because of the availability of the phenyl or the tryptophans, right? Does that make sense?
Yeah.
Now, there's something else involved as well, though. Let's zoom in here, see if we can see some of these tryptophans and tyrosines. Lots of tyrosines here. Where's our tryptophan? There it is. There it is. Okay, so here we have a tryptophan and here we have a tyrosine. Let's imagine that this tryptophan here, which is kind of buried, but let's imagine that it eventually gets some electromagnetic energy hitting it, and then it re-emits or fluoresces some photons of energy in all directions, okay? Well, that energy, as it's being emitted this direction, has a likelihood of being absorbed by surrounding molecules, and specifically by something like a tyrosine, okay? So again, we have another sensitive component in terms of fluorescence to tryptophan's proximity to other fluorescent, or even if they're slightly, even significantly less fluorescent subgroup or residues, right? Side chains. So that's another parameter that will change every time the protein moves. Now, in general, the protein is kind of just always constantly moving, right? And you get an average of all the fluorescence. But if you do something that causes the protein to change significantly, then it'll, be moving still, but the fluorescent signal will shift, okay? Proteins don't always unfold so neatly, kind of like I'm showing here, all right? Sometimes protein folding and unfolding, or specifically unfolding. So here I have a simple protein that I've drawn, and we'll say that's folded. Well, when we unfold it, we'd like to think it forms a nice straight chain when it's unfolded. But that's kind of an unrealistic way of what's actually happening. When we get unfolding, usually it's something like this. It's a different kind of mess that is no longer functional because it's not the correct fold. All right? So oftentimes you can get and that's what we're going to be looking for in this lab, is a change in fluorescence due to unfolding of protein. Okay, and that's kind of like what we saw in that last lab, is that as you increase the concentration of dimethyl sulfoxide, the catalytic ability of the myoglobin decreases, and that's because the DMSO is unfolding the protein. The interesting thing about that, though, is that even with very high concentration, you still have myoglobin that can react well. And, industrially, there's a lot of chemicals that we would like to specifically oxidize. And protein oxidation at an industrial level is not unheard of. You can train proteins or make find proteins that are very good at oxidizing specific parts of other chemicals, but sometimes those chemicals are, pharmaceuticals that are not soluble in water. A lot of pharmaceuticals aren't very soluble in water, and so to perform those reactions, you need to have something to help. And DMSO can do that. DMSO is an organic solvent that is soluble or miscible, we say, with water. Miscible is when you take two solvents and combine them together. And that results in a solution, DMSO, 50% DMSO, 50% water, that you can begin to dissolve lots of hydrophobic molecules in, hydrophobic compounds in, and therefore they're available to perform chemistry on. If you just put some powder that won't dissolve in the water and the powder goes down to the bottom, it's not going to be able to react. You're not going to get any reaction. Okay, so unfolding. We're going to monitor fluorescence as we unfold a protein, and we're going to add a denaturant. The two most common denaturants are called urea and guanidinium hydrochloride, or guanidine, right? These denaturants function I mean, well, in terms of unfolding, but you need to have a very high concentration. So we change the solution concentration of something like urea from 2 to all the way up to 8 or 9 molar urea, which is very high concentration. And now, do you know the concentration of water in water?
100%?
No, I mean the molar concentration. What's the molar concentration of water in water?
1 molar?
No. So let's think about this. I want to know how many moles per liter, moles of water there are for every 1 liter of the solution. It's not really a solution now because it's just all water, but we can still calculate the molarity. Okay. You know what we can start with to solve that problem?
I guess I don't.
Well, we can say that we know there are 18 grams of water for every mole of water. Is that right?
Oh, okay, yeah. But don't you have it upside down?
Oh, right. Yeah, yeah. So 1 mole of water, and that's good because it's on top now, is 18 grams of water.
Well, now what are you going to do?
We need to get rid of grams of water, right, and get it to liters.
Well, you can't do that.
Well, you can't go grams to milliliters, right? What do you call that?
Oh, that's the density. Yeah, 1 gram per milliliter.
1 gram per milliliter. Very good. So I can go 1 gram per milliliter, and then I can go 1,000 milliliters for every liter. There we go. So I got moles per liter. So 1 divided by 18 times 1,000.
I did it on my calculator. It's 55.
Okay, 1 divided by 18 times 1,000. So 55.5 molar water. All right. So if I have a container here, and now the water, that means there's 55 moles for every liter, right? And we always talk about molarity in terms of concentration. You know, if you look at a little portion of your water here, this is just, I'm looking at a little portion there. In that box, the ratio of chemicals are, if this is, let's say, 1 molar NaCl, right? Just 1 molar solution of sodium chloride. That means I have 55 units of water for every 1 unit of sodium chloride, right? And that sodium chloride is busy is being, surrounded by water molecules. The sodium ions are surrounded by water molecules. The chloride ions are surrounded by water molecules, right?
Yeah, because it ionizes.
That's right. If something dissolves in solution, it's because the water molecules are busy surrounding it, okay? Well, when you make a solution that becomes 10 molar in something, then that keeps the water molecules busy and begins to distract them from solubilizing other chemicals. The reason why proteins fold is because they're in an aqueous solution. Or another way of thinking about that is the aqueous solution provides the forces necessary to drive this folding. If you put a protein like this in something like ethanol or acetone, right, this won't fold. This won't fold that way because the molecular interactions with water molecules are gathered all around this thing, right? And it is those forces between here, water molecules and your protein, that's driving that protein to fold the way it does. A lot of these residues are hydrophobic. Remember what hydrophobic means?
Afraid of water.
Yeah, or that they are not very soluble in water, right? Alanine, isoleucine, leucine, methionine, right? Valine over here, tyrosine, all of these ones are hydrophobic side chains, right? So these side chains are going to kind of act like a drop of oil in water and clump up and try to get close to each other to get away from the water. Whereas ones like these, and even these ones up here, these charged ones, or these polar ones, right? Hydroxide groups and amino groups here. These are very polar, and they want to come out and interact with the water molecules. So it's these forces that drive the folding. So when you add urea, one way of thinking about what it's doing is it is occupying some of these water molecules, pulling them away so that there's less interaction with the protein. The water molecules have less time to spend with the protein, and then that causes the protein to slowly unfold. So we're going to see if we can monitor this unfolding by monitoring the fluorescence. Now, the protein fluorescence shift based on a protein's unfolding is not always predictable. After you've run the experiment once, then you know. But sometimes the protein will increase in fluorescence when it's unfolded, and sometimes it will decrease in fluorescence when it's unfolded. Now, if a protein increases in fluorescence, what are some reasons why that increase in fluorescence might be occurring when it unfolds?
The tryptophans are more available to absorb and emit light.
Very good. So unfolding, the tryptophans might be more available. What are some reasons why you might see a decrease in fluorescence when the protein unfolds?
I'm not sure. I don't know if I can understand that one.
Well, you could get a shift in such a way that the tryptophans actually get buried more, or the tryptophans come in a closer proximity to other fluorescent molecules or other molecules that can absorb the light, the electromagnetic energy that the tryptophans are fluorescing. Okay, so tryptophan, tyrosine, alanine, phenylalanine, tyrosine, and phenylalanine, tyrosine. Yeah, those are our major other fluorescent contributors, right? They can absorb energy as well. So now, protein unfolding is kind of catastrophic, we say. Or in other words, if I have something here like a measurement to predict unfolding, as I add or make my perturbation, whatever it is my unknown is, maybe it's going to be concentration of urea, right? A chemical unfolding. This can be temperature. There's a variety of different ways that you can perform the unfolding. But if I'm going to monitor unfolding, and maybe it starts up here in terms of the amount of foldedness. So this would be 100% folded. This would be 0% folded. Now this could be monitored by fluorescence or whatever. But there might not be a whole lot of shift until you get to a point where, because it's being bothered and perturbed enough that it all of a sudden collapses, and so you get a shift like that. All right, and so this is a sigmoidal curve, and it doesn't have to be down; it could be up as well. But the point is, it happens kind of all at once, so it's cooperative, we say, because little parts become unstable, and then once the instability reaches a point, the whole thing collapses and unfolds. So this is the sigmoidal curve is a typical unfolding curve. Again, could be going up or could be going down. Typical unfolding curve for protein unfolding. Okay, so let's look at the methods that we followed for this experiment. So this is how this experiment was performed that we're going to use the data from. We used propanol. Propanol, you've heard of isopropanol?
Yeah, that's in the medicine cabinet, right?
That's right, that's rubbing alcohol. And we increased the amount of propanol as we decreased the amount, decreased the amount of buffer here. All right. And the amount of protein stayed the same. This was myoglobin. We use myoglobin as a protein here. And these were the numbers of the molarity. This is the molarity of the propanol, 1 through 5 molar. And then we used a fluorescence spectrometer, and the excitation wavelength was 275. What do you think that means?
Was that the wavelength that it emitted? Oh, no, it says emission wavelength. So that must be the wavelength that it absorbed.
That's right. 275 nanometers. Is that in the visible region or the ultraviolet region?
That's in the ultraviolet, right? Because it's less than 400.
Very good. And then the emission wavelength?
Oh, 400 nanometers.
Okay, so we're monitoring emission at 400 nanometers. And we have information about the slit widths. Do you remember what the slit widths were?
No, I don't remember.
Well, in a fluorescence experiment, because the light, the different kinds of, the different amounts of fluorescence can change significantly from sample to sample. You can get intensities of only 1, 10, 20, something like that, but you can also get really, really high intensities. Our way to control that is to either decrease the amount of absorbed light by, this is one of our slits right here, decrease the amount of light that can come in, and then as the light goes off in all directions, we have our detector here. We can also change that slit width there. Okay, so those two slit width values are important to know exactly what is going on. So they were at 10 nanometers. All right. Very good. And you can read through one other important thing that I'll note here, you can read through that and you can pretty much copy and paste that into your methods. But a four-sided cuvette. A four-sided cuvette is important. Well, any ideas why you'd have to make sure you used a four-sided cuvette? I don't know if you're familiar with the difference between a four and a two-sided cuvette.
I remember in Chem 105 we had the cuvettes and they were like frosted on two sides.
That's right. They were frosted, or they were kind of hazy. Because, do you remember what we said, why that was?
That's where you could touch it.
That's right. But a fluorescence cuvette can't be that kind of cuvette. The fluorescence cuvette has to be clear on all four sides. Any ideas why?
Well, because you're detecting at 90 degrees.
That's right. You're detecting at 90 degrees, and so the all four sides need to be transparent. Okay, very good. Another thing about this cuvette that's probably isn't really mentioned here, but well, I guess nowadays things have changed a little bit. But in the olden days, and in some cases, you still have to be aware of this. Glass cuvettes and plastic cuvettes will absorb ultraviolet light. So 275 nanometers is ultraviolet light, and so you used to have to use what's called a quartz cuvette. Quartz cuvette, which is a glass that's sintered a specific way, will not absorb UV light. But plastic, glass, it will absorb UV light. Now, what's changed is we've been able to manufacture our plastic and mold it in such a way that it now is UV transparent down to a certain point. Not completely, but down to a certain point. And usually it's enough that we can use them. So that's another thing that's important to note in terms of the kind of cuvette that you use when you perform fluorescence. All right, so I'm going to provide you the signal for these different results, and we're going to plot the signal versus concentration. I'd like you to look up protein unfolding experiments using fluorescence. You can look up protein fluorescence experiments using myoglobin, using heat, right, as a source of unfolding, using urea as a source of unfolding, and let's compare it. Let's see if our data is comparable or if we're just looking at some anomaly. All right. Yeah, those are the things you want to discuss in your results, and you're going to want to find a paper that talks about that. Okay, great.